Hi, we are back again, and in the previous session I did a brief course on brain imaging, various modalities, the principles, everything crammed into one. I feel that is sufficient for the audience. People who are interested are definitely welcome. It is a very interesting field of study, and reason I had to delve into it is because what we are going to discuss now is the output of the whole story, and so you need to know how the input is to actually figure out how to interpret the output. So that was the point. Now we will start with a brief revision of some keywords. So brain imaging. What I am going to discuss is going to be MR images because of its high resolution and popularity with students and for various kinds of research projects. So that's one reason why I am doing. You have to understand that these are tomographic graphic, basically indicating that slices of tissues are interrogated, and you would need to know so these MR images are available in something called as DICOM format. So these are the these are keywords which you need to understand. So slice based imaging also indicates that you should know what is axial, coronal, sagittal imaging, and so these are terminologies which you should be familiar. So when we spoke about resolution, we spoke about slice thicknesses of resolutions, overall resolutions of 0.6 mm resolution between two structures. It is nowhere what as surgeons we require, and as many many people who do diagnostic imaging require, but it's the available resolution. So tomography is you are interrogating tissue in slices. So when I did my anatomical description, I made it a point to show you stuff in 3D and in best fashion which I could think of. But whenever you are looking at any kind of imaging, you are always looking at slices. There are very few places where you actually look at 3D. That is because actually of the way in which the imaging has developed. So, for example, we as surgeons, we would prefer to see everything in 3D rather than 2D, because it takes some effort on our side to interpret a 3D image which you see on CT and MR images and films and stuff, and then assimilate the 3D knowledge out of it. But anyway, that is how it is, and you cannot change things. DICOM is important because that is the mode of communication. It is the standardized format in which the images are given to you in slices, and then you have a DICOM viewer which is necessary for viewing the images. DICOM images usually are given along with the CD, pen drive, etc., and then you need to open. There are several open source DICOM viewers which are available online, through which you can view the imaging. So that is about it, and then you need to understand these terminologies. What is axial images, coronal images, and sagittal images. So axial is like this. Okay, so it is a slice. So your entire head is in slice, and that is the axial images. Coronal is in the coronal is like this, parallel to your face, so as to say. Sagittal is like this. So coronal, axial, and sagittal. So that is the terminology. We no need to mug up this, I will be showing it to you, but I needed you to be comfortable with the terms rather than what actually they mean. So that is the idea. Now if you look at imaging, this one you would find there is an app which runs the DICOM viewer, and then you can basically scroll through some kind of imaging which is available. So that is how these things run. Okay, so anyway, I am not going to show you that part of the story. What I would want to make you see is how to interpret images, and I use this particular thing for demonstrating my images. So, this is from Invest which is one of the packages. There are several of these packages. So, there is ITK Snap SNP, which is one of the most popular ones. You have Osiris, which is a viewer platform on Apple, and my personal favorite is Invest for the kind of research stuff which we do for some specific purposes. So, I am using Invest because I am comfortable with it, and I can show you things which are there. So, so this is a coronal image. I can draw. Okay, so this is what I mean by a sagittal image. The eye in front, this is the back of the head, upper part superior, inferior. So, this is a sagittal image. So, that is how a sagittal image looks like. This is an axial image, it is upside down. I will be sorting it out in the description part of the story, but do recollect. So, this is how the eye is on one side, this is the eyes, two sides eye and the back of the head. This is coronal section. What you can make out is the ears. So, we will start our run of anatomy through the axial images. So, this is how the image looks like. We will, so there is something called windowing. So, windowing helps in making things. Windowing is a method of changing. It is a concept which is basically borrowed from CT, and what windowing does is it helps you to improve the contrast, and there are two things, windowing width, and this is the larger window which you can change. Looking at an optimal place where you can see. So, now, I have not spoken to you about MR sequences. MR sequences is something which I have to speak about. I am not sequences. Now, I spoke to you about how hydrogen you give the magnetic field and then you give a radio frequency pulses. So, combinations of these parameters, radio frequency parameters, the magnetization parameters are given as user controls, and that is how you have various sequences such as T1, T2, then you have flare, then there is cis image, then phase, then contrast. Contrast is you give by injection some material into the blood which is gadolinium, and that shows up. It's relevant for especially structural lesions such as tumors, in which the tumor takes up the gadolinium and helps in contrast differentiation from surrounding brain which does not take gadolinium. So, so these are there are several other kinds of sequences, but what you would need to remember these are these set of things. So, sequences determine the way in which you interpret brain images. Each sequence has its relevance, it has its medical relevance. There are certain things which are better seen on a particular sequence, and that is the point. But if you remember, I had used the analogy of car gears for that purpose, because they are fixed. You have only those parameters which you can tune, and even people who are doing image processing have to remember what sequence is being used, because you use an inappropriate sequence, you do not get the relevant output which you want. So, please be careful in choosing sequences and choosing sequences for your study, for your analysis, for your data, whatever. So, they all these sequences image the brain, but they look at different issues in the brain. So, I have not included fMRI, then diffusion tensor imaging. These are more advanced techniques. Each has, as I told you, each has its relevance. fMRI, for example, is functional. So, it tells you something about the functional activity which is there in a particular part of the brain based on oxygen consumption, and it is unfortunately of low resolution. So, the functional MRI is superimposed upon normal MRI. Functional MRI itself is a derivative of statistical analysis of very fast acquisitions. So, you have multitudes of images taken in a short period of time, then you statistically average to get the output. So, there is a lot of statistical algorithms which are there which get you the fMRI images, and the fMRI images in turn are superimposed on T1 sequence images. So, please do look back into that. So, when you, if somebody is starting with saying, yeah, I am going to study fMRI images and that is the output image which you are seeing with all the colors and stuff like that, please remember that it is not, there are two base imaging sequences in play. The acquisition parameters of both the imaging sequences are completely different. The purpose of the acquisition is different, the slice thickness is different, everything is different. So, what we as clinicians see and what you would do in image processing should be completely different. Please do not use the same perspective which we have for processing these images, that is the point. So, we will come back to our, I am not teaching MR imaging over here, I am teaching anatomy over here. So, we will start with some basic notions which so this is a T1 sequence. It is actually not a complete T1, it is called a highly weighted T1 with a specific purpose. It is designed so as to give you idea of gray and white matter tissue. Okay, so that is the idea of this sequence. What is obvious here? We will start with the obvious and then go to other things which are not so obvious. So, this is the eye over here. So, please remember this is left, this is right, anterior, and this is posterior. So, that's the nomenclature which is there. So, anterior is eye and sectional indicates that it is a section. So, section is the eye is the most prominent part. So, between the two eyes is the nose, that is the nose area. So, that is to give you a context of how things are being described. So, we go, this is we are going up, scrolling up as in towards the scalp and above the eye. So, this is there is no eye seen over here. So, that is how we scroll above. This is an axial slice, remember again. So, axial slice we okay. Now, we have reversed movement going down to see the eyes back again here. These are the two eyes again. We continue down to see the nose. This is the paranasal sinuses on either side. This is the nose again still. This is the teeth. Okay, so teeth, jaw, teeth, lower, meaning the lower part of the teeth, and so that's the lowest most part of the structure. So, we look at other parts of the anatomy, anterior, posterior. Generally, we would see it like this, flip. So, anterior, posterior, this is sagittal section. So, sagittal section is vertical across. So, this is an eye again. Please do not look at the nomenclature here, I have done a flip. So, this is the eye, then this is the back of the head, upper part of the head, lower part of the head. Coming towards the midline, you can make out the nose here, tongue here, and then the opposite side of the brain. So, this is sagittal. I am repeating so as you become familiar with the terminologies and what set of images are associated with that. The last kind of sequence is the coronal sequence. So, coronal sequence is side to side. So, how do I show side to side? I should show back the eyes and then you would get an idea. So, this is side to side cut and so here comes the eyes. Here are the two eyes. So, this is eye and nose, and you should always interpret images as if the person is looking towards you. So, when the person is looking towards you, this is the left eye, right eye, and that is how it is. Okay. So, these are conventions, there is no hard and fast rule. In fact, you saw me flip through the image a couple of minutes back. So, that is how it is. So, now once you have got a fair enough orientation of what axial, coronal and sagittal means, I will start with something again which we know. So, in my 3D demo, I had spoken to you about this separation which is there between the two halves of the brain. So, here you can see the structure, that is the fox. Okay. So, this is one hemisphere, this is another hemisphere. So, I told you a person is looking towards you. So, this is the left, this is the right side of the cerebrum. I am using the term cerebrum because it is the upper part of the brain. So, we go back, cerebrum, 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 we can see the fox continuing. Then we shift down to understand this part. Okay. So, here you can see some structure white over here and white over here. If you remember my 3D discussion of the skull, that is the tentorium. So, the tentorium divides the cranial cavity into two parts. These are sections. So, sections, continuum sections. So, they are the tentorium separates the cerebrum, some cerebellum, splits the skull into supratentorial and infratentorial. So, here we can see the differences between the two parts. This is the cerebrum, note the architecture of the cerebellum. So, this is cerebellum, cerebrum. Fine. So, generally we would discuss anatomy in the context of axial sections. So, axial sections are the primary way in which we would discuss anatomy. So, we start with something which are obvious. So, this is the cerebellum. Cerebellum. So, this whole stuff is the cerebellum. Two hemispheres like in the cerebrum, you can make out very clearly. These two small structures over here are called the tonsils. Tonsil, tonsil. We come up a little bit and this thing is the uvula. So, these are things which I shown in my 3D anatomy class. So, the same things in 2D how they look. So, this is uvula, uvula, then these things down, these bulges here are the tonsil. This is cerebellar hemisphere, this is cerebellar hemisphere, this is the left cerebellar hemisphere, this is the right cerebellar hemisphere, it is by convention. So, and this round structure over here is the brainstem. Not very obvious, see, which is one of the reasons I did not want to start teaching anatomy through sectional imaging. Sectional imaging is advanced imaging because you need to understand what is there in 3D before you actually interpret what is there in 2D. So, this whole stuff is the brainstem, and you can make out that there are no distinct boundaries between brainstem and cerebellum, because the brainstem and the cerebellum communicate through things called as the peduncle. So, this is the communication between the brainstem and the peduncles. Now, again going back to what I had taught you earlier, how do you recognize various parts of the brainstem? Yeah, so this is lower down. So, we are again coming down, this is almost the neck, this is spinal cord. So, this white stuff is the spinal cord. You can imagine that its resolution is not adequate in spite of the kind of imaging technique. You can see the pixelations happening at this magnification. These are very high resolutions. In fact, the reason I am using this imaging is because it's been acquired using the best possible techniques for white gray differentiation. So, brainstem. So, here lower down spinal cord continues into the brainstem. So, if this is the brainstem, then what part of the brainstem? This would be the medulla, medulla which continues down as the spinal cord. So, anteriorly would be the midline small deflection which is the fissure. On either side, these bulges which are seen are the pyramids. Okay. So, these would be the pyramids again, yeah, now you can see this transition. So, it is jumping and becoming bigger. So, this whole part would become the pons. This is the pons up to here. So, pons is the place where there are connections with the cerebellum. If you go back, there are no distinct connections with the cerebellum here. You can see that there is a boundary here and then there is a clear boundary here. So, when it comes to the pons, it is obvious that the pons communicates with the cerebellum. So, these are through something called cerebellar peduncles, superior, middle and inferior. So, this is the cerebellar peduncle. We come up, this is still the pons. Posterior fossa, and this white stuff which is seen is the petrous bone. Yeah, so I think this structure should be familiar to all of you by now. So, this is the midbrain. So, we started from below, we come up from medulla to the pons and to the midbrain. Midbrain, you can see small bulges behind, this one bulge here and this one bulge over here. So, they are the colliculi, right, and so that is the that would be the superior colliculi, which is the bulge over here, and this should be the inferior colliculi. So, that is how the colliculi, and that is the midbrain. Midbrain, pons and lower down medulla, cerebellum is here, left hemisphere, right hemisphere. So, axial sections starting from below, cerebellum, medulla, tonsil of the cerebellum, the hemispheres of the cerebellum and the medulla. Coming up, you can see the uvula come into focus, which is the central part. This upper part, the lower part is the uvula, but the upper part is called the vermis, which connects both the cerebellar hemispheres. So, this is the vermis, cerebellar hemisphere. Then we come to the pons, and coming up, yeah, that split which happens. So, this is the pons and then suddenly we are in the midbrain. So, midbrain still part of the cerebellum seen. So, this gap through which the brainstem and the cerebella is called as the tentorial incisura. So, opening in the tentorium, the tentorium in turn splits between the upper part, which is the cerebrum, and the lower part, which is the posterior fossa containing the cerebellar hemispheres. Another structure seen black in color. So, this thing is the ventricle. So, which ventricle it would be? It would be the fourth ventricle, because we are starting from the base and coming up. So, that is the fourth ventricle. So, what happens to the fourth ventricle? We trace it up and just above you see that it becomes a small cavity over here, it is called the aqueduct. Right. So, what happens above is the cerebrum comes into place. So, we go back from below. On either side, the first structures which you see almost in relation to the eye are the temporal lobes. So, this is the left temporal lobe, right temporal lobe. Yeah, so this is another place. So, this would be the frontal lobe, parietal lobe, occipital lobe. Another thing which I would like you to appreciate is this small gap here. Small gap here, and that gap is the insular cortex. Same on the opposite side. So, insula is surface which is buried under the brain. So, there is brain tissue here, there is brain tissue here, and in between is this thing called as the insula. And so, that's the parts of the cerebrum from the outside. When we come up, we can see the clear separation between the two cerebral hemispheres. This is the beginning of the fox which separates, it is the dura which separates the two hemispheres, and then there is this black gap in between which is the space between the two hemispheres. We would be seeing the corpus callosum. So, in a axial section you would see the corpus callosum both in front and somewhere in behind. So, this is the splenium of the corpus callosum, this would be the genu of the corpus callosum. So, that is about what all we can see. Internal structure and the other anatomy we would go in the next phase. So, these are structures which are seen on the axial images of the brain. So, what would these be? Empty spaces within the brain are the ventricles. So, ventricles, that was the lateral ventricle here, which is in the cerebral hemisphere. The small place here in the center is the third ventricle. So, the third ventricle actually goes down into the midbrain, which is here, midbrain is starting over here, and this would be the aqueduct. The aqueduct opens into the fourth ventricle. So, lateral, third and fourth ventricle. So, that is the nomenclature. Communications are there between each of these ventricles, and that is what you would need to remember. Okay. So, for the next part of the story is the deep gray. So, deep gray is I had highlighted that this is the So, deep gray is present within the center of the brain. So, center of the brain, it is closely related to the ventricles. To highlight the deep structures, I think I need to change the windowing a little more. Yeah, so right. The first structure which I want to teach is about the thalamus. Two thalami on either side. So, this is the egg like shape structure which is over here. It is thalamus is almost in continuation with the midbrain. So, when you come up, this is the midbrain, shape is very obvious, two cerebral peduncles, aqueduct behind. When the cerebral peduncles come up, you would find the thalamus, it is obvious over here. So, you can find out some structure here, darkish. So, this is white matter, this is grey matter. So, that is the differentiation. So, white and grey, this is the thalamus in here, thalamus in here. This bulge which is coming into the ventricle is the caudate head. So, thalamus, caudate head, and the remaining structure is the lentiform nucleus with the globus pallidus which is inside. So, that constitutes the deep grey, and that also sort of completes the discussion in the axial section. Let us see how these things look in various other sections. So, once the axial section is understood, we proceed on to the coronal section. So, coronal section, we since we started from behind, we go back. This is front to back. So, this is the tentorium. So, tentorium separates the cerebrum from the cerebellum and the brainstem, of course. So, the boundary is obvious, it is not obvious on imaging. So, please this is one of the fallacies with current imaging, though there is good resolution, it is nowhere near actuals, and it is with actuals that we deal when you are actually treating patients and people. So, there is lot of gap between what is seen on imaging and what is actually present, and these are not mythical structures, they are very well very well nicely seen, but unfortunately on imaging with even a 0.6 mm resolution we are not able to differentiate these things. It almost looks like there is no boundary between the between the cerebrum and the cerebellum. So, here again we start from this, we are going from back to front. Okay. So, I have shown you, I did not describe anything in from back to front. So, this is the left cerebellar hemisphere. Each of these frond like structures, you can make out that it is looking like leaves of a tree. So, each of this is the cerebellar folia. So, the foliar architecture is very nicely seen, and you can make out this foliar architecture over here. Somewhere anterior would be, there is a grey area over here and a grey area over here. So, those are the deep nuclei of the cerebellum. So, cerebellar cortex, this whole thing is cerebellar cortex, white matter of the cerebellum, white matter of the cerebellum, white matter of the cerebellum. So, this whole thing is the cerebellar cortex. Remaining, remember that this is a coronal section of the brain. So, coronal section of the brain with these structures. So, this thing over here you can make out is the uvula. It is seen differently on this kind of on the coronal section. Uvula is the center, is the side structures, and this is the tonsil. So, tonsil continues into the vermis. So, how do I show the vermis? Yeah, so these things are the vermis. This. It is quite different from the hemispheric structures which is there, which is white matter in the center and grey matter in the periphery with the cerebellar folia. So, cerebellar hemispheres, then this is the connection. So, this is the brainstem, connections to the brainstem, these white structures are connections to the brainstem. The central thing is the brainstem as such. I will reduce zoom. spinal cord, medulla, there is this bulge which is coming over here, which is the pons, and upper part is the midbrain. Not very obvious as it was in the axial sections, but it is there. So, you can make out the size discrepancy between the two, which I tried to highlight in my 3D model. So, this is the medulla, the bulge of the pons, and the midbrain, which is somewhat bigger than the medulla. So, when we come anterior to that, this would be the aqueduct, third ventricle and lateral ventricle. So, we see a lot of structures in this. This is the lateral ventricle, again cut section, please remember, this is the third ventricle, and lower down is the aqueduct, which is connecting with the fourth ventricle. So, fourth ventricle is this black space. So, black space, not very clear on the axial imaging, but here you can make out that it is a distinct cavity, and the very distinct cavity continues up to the aqueduct. So, the aqueduct and then continues into the third ventricle, and then through the third ventricle it goes into the lateral ventricle. So, we go further up, cerebrum. So, front you can make out this distinct separation between the frontal lobe and this is the temporal lobe. So, temporal lobe downwards, frontal lobe upwards, and there is the Sylvian fissure which is over here. So, that is how it gets differentiated. Not very clear here is the insula, which I think I should showcase. Yeah. So, this is a better delineation of the insula. Yeah. So, within the deeper parts of the brain, so the deeper parts of the brain is this structure, this is the insular cortex. Temporal lobe, frontal lobe, and posteriorly this is the parietal lobe, temporal lobe, insula. So, that is how it is. So, this space here is the Sylvian fissure, this is the insula, and these would form the ventricles, lateral ventricles, this forms the third ventricle. Now, so we spoke about thalamus in the axial section, let us try if we can get that in the coronal sections. So, we go back, change the window to appreciate the gray matter better. So, this is the brain stem, this would be the midbrain area, and then just above the midbrain area, this round structure over here is the thalamus. Come more front, thalamus is only a small part over here. This is the caudate head which projects into the ventricle, caudate head which projects into the ventricle, lentiform nucleus, a bit more clear, and I think this anterior part is the globus pallidus over here, caudate head. So, this is caudate head again and the lentiform nucleus. So, that sort of finishes the description in the coronal sections. Sagittal sections, sagittal is from front to back. Okay, again to make it clearer, this is the eye, one eye, you can see a small structure which is coming out from the eye, that is the optic nerve. Yeah, so these are things which are to be appreciated over here. So, we have so far just discussed about the corpus callosum. So, here you can clearly see the corpus callosum, I will zoom it further. So, rostrum, genu, body and the splenium. So, that is how the that is how it looks like. This whole stuff is the corpus callosum. The corpus callosum connects between the two hemispheres as the biggest connecting fibers between the two hemispheres. We actually do not know much about the purpose of the corpus callosum, but operatively we know that if we can cut about 2 centimeters without causing any severe problem. So, the corpus callosum communicates between two sides. Now, in my 3D description I spoke about the fornix. So, this is the fornix over here right underneath the corpus callosum, and it is going from front to back, and it goes to this thing called as the mammillary body. You can't see it in a single structure, but single section, but you can see that it is this is the fornix, and then it goes around the thalamus. So, this is the thalamus here, brainstem here. So, this is the midbrain. You can see the midbrain which is continuing almost with the thalamus. Just below the midbrain is the pons, which you can again see as a bulge, a distinct bulge which is separate from the midbrain. So, following the midbrain following the pons down, you can see this pons, and there is another bulge over here. So, this is the pyramid of the medulla, and the pons ends here, pyramid of the medulla. This part is the floor of the fourth ventricle. So, floor of the fourth ventricle, I took some time to explain how it is rhomboid in structure. Then there is the roof of the fourth ventricle which is formed by the cerebellum. So, this is the V shaped roof of the fourth ventricle, this is the fourth ventricle, and this is the foramen of Magendie in the midline. So, this is midline. So many things in the midline which we did not see in any of the other sections. So, this is corpus callosum, fornix, midbrain, pons, medulla with the pyramid, and this is the spinal cord. I go a bit outside, that is lateral, and you can see the uvula, this is the tonsil, this would be the uvula in the midline. Okay, and we go much more further, only things which you see are the cerebellar hemispheres. Cerebellar hemispheres, you can see the white matter in the center, the white grey cortex in the periphery. Coming up, you go back to the center. So, this is the center because we are seeing the corpus callosum as a distinct identity. You do not see it in continuity with something else. So, this structure is the tentorium. So, the tentorium separates the supratentorium from the infratentorium, cerebrum from the cerebellum, and through the incisura, which is the opening in the tentorium, the brainstem and other structures communicate with each other. Yeah. So, so once that is the splenium is understood, what comes underneath is the lateral ventricle. Then this is the communication, this is the region of the communication between the lateral ventricle and the third ventricle. Third ventricle is again in the midline. So, this black space over here, third ventricle, this is lateral ventricle, communication through across the foramen of Monro, which is situated over here. So, we will see if we can trace out the aqueduct over here. So, this is the aqueduct here, central space communicating within third ventricle over here. This is the fornix. So, third ventricle here, third ventricle, and through aqueduct into fourth ventricle over here. So, that is how it is arranged. Then we see if we can identify further structures difficult in the sagittal structure. sagittal section. So, there is one structure which I think is very obvious over here, which I should highlight. Yes. So, you can see something over here. Now, this is the temporal lobe. So, this is the frontal lobe, parietal lobe, occipital lobe, temporal lobe. In the temporal lobe is the continuity of the lateral ventricle, and that is the horn, and then this is the hippocampus. So, you can see one structure here which is projecting into the temporal horn, and that is the hippocampus. Right. So, the hippocampus curves around the thalamus and forms the fornix. So, this is the fornix. Can't make out very clearly where it is curving around, but then you go laterally, you would find that it is going into the hippocampus. Now, just above the hippocampus is the other structure which I wanted to highlight. So, this is if this is the hippocampus, sectional anatomy. Please remember, this is sectional anatomy. This gray structure over here is the amygdala, and the amygdala is closely related to the lentiform nucleus over here. So, go back into the ventricle. When we go back into the ventricle, this is the frontal horn. There is another gray structure over here which is here seen over here. So, this gray structure is the caudate nucleus projecting into the ventricle. You can see that it is distinctly projecting into the ventricle. This thing is the caudate nucleus. So, head of the caudate nucleus is just about somewhere here, and that is how it gets. See, again you can't make out distances here because they are in different sagittal planes. So, that is how the marking is, and we need to identify insula. So, insula is this. So, you can see white matter all around. So, it indicating deep within the surface, within the brain matter, and suddenly you see gray matter over here. So, gray matter with cortex sulci and subarachnoid space. So, this is subarachnoid space, subarachnoid space, subarachnoid space. So, that is the insula. So, that is about how you interpret structures. These are very broad structural descriptions, but it is to give you an idea of what is situated where, okay, and how these things look like. I tried showing it to you in 3D, now I showed it to you in axial. I will try showing it in another sequence. So, this is a T1 sequence. I will be showing you the so, this is something called as a cis image. So, cis image, again it is a gray scale image. What is best in this image is that you can make out structures which are outside and inside of the brain. Okay, so, that is the idea. So, if you recollect from my earlier discussion, I spoke a lot about the structural anatomy without delving into the gyri, sulci, nothing of that discussion was there. So, that is being described over here, and we will see how best to make use of this image in understanding about the gyri and sulci. So, gyri and sulci are now very obvious. You can see so many gyri over here, and each depression is called as a sulcus, and there are so many sulci around here. So, how do we make sense of an image like this and try to map our 3D knowledge which we acquired earlier and put it on to this. So, for that reason we will start with notions which we had already solidified, concrete ideas, and we take up that. So, this is axial section. So, when I say axial section, there is left, right, anterior and posterior. How do we make out anterior and posterior? We scroll through that, confirm our anterior has anterior part has eyes, so that we do. So, these are eyes obviously, and you can distinctly make out the optic nerves. So, you can see the optic nerve over here. So, this is the optic nerve, and you can see it is pretty lazy as in loose, and that is because to accommodate for the rotations of the eyeball within the socket. You know, the nerve should not get stretched, it does not get stretched when you look at towards one direction, and that is the purpose of the, that is the optic nerve. This is the eyeball. This black thing is actually the lens, this is the nose. So, orientation done, we go back to our primary description. Axial section. So, we can scroll above and below to find out. So, I am going above. How do I know that I am going above? Because I am still seeing cerebrum and I am not seeing brainstem. So, if I am going down, I should be seeing brainstem. So, as opposed to the T1, you can distinctly see this gray line over here, which is splitting it into two halves, and this gray line is the Fox cerebri, FALX cerebri. So, Fox cerebri. Now that we have identified the Fox cerebri and said that this is the left cerebral hemisphere, right cerebral hemisphere, and we have acknowledged the presence of gyri and sulci, we need to look at the midline. So, midline has all this swelling. So, gyri one, then there is a sulci here, and then there is a that is a sulcus here, and then suddenly we dip into here and then find that this sulcus is going all the way through up to the lateral part. So, that is a key differentiating discovery, and it is important because that we, by that we have identified the central sulcus. Now, I told you that it may not be easy in everybody to identify all these structures. So, we need multiple markers of the central sulcus, and that is the purpose of showcasing this image. Now, what are the other markers of the central sulcus? The central sulcus is important, reiterating stuff which I have already highlighted. The central sulcus divides the brain into an anterior active part, which is the motor part of the brain, versus a posterior part, which is the sensing part of the brain. Now, they are not strictly rigid in terms of acting part, because acting as in functional function as in implementing part. Implementing includes judgment, decisions, thought, everything is an action, and so that is what is happening in the anterior part of the brain. So, going back to the image, this is the central sulcus. So, what comes anterior to it? What is the gyrus anterior to it? You can see it is thick. This gyrus is thick when compared to this gyrus over here. Same thing on the opposite side, thick gyrus, thin gyrus. So, the thinner gyrus is the sensory cortex, thicker gyrus is the motor cortex. Number 2. So, reiterating, the sulcus which goes from the center all
Just 5 seconds.
That is okay, no issues. Anyway, it is 59 minutes, you can reset it, another half an hour it will go on.
No, sir, you can just end it up after this one.
I know, I will, I am ending it and then restarting it again.
Okay.
Yeah. On, no, this is.
Yes, sir. No, no. I'll come and reset it.
It's okay. No, in the sense that I am ending and restarting in the same this one, because it is a continuum. There is a lot more stuff which has to be told.
You can start, sir.
So, that is the central sulcus, thicker motor cortex and the thinner sensory cortex. You have to come up to actually find out where the where the sulcus starts from. So, if you go down deeper, you would find that the same sulcus is only on this side and it is not communicating over here. So, that is a key step. So, we will break here and then continue along with this. So, we have identified midline and how to identify structures on the surface of the brain just using imaging, brain imaging, to find out the what stuff I had taught you in 3D on the 2D. Okay.